So just what is this term called glycoimmunology? Glycoimmunology encompasses bonded sugar molecules known as monosaccharides, polysaccharides, or oligosaccharides, etc., which are basically carbohydrate-based structures, also known as glycans, all of which are intimately involved in glycoimmunology. In fact, the loss of any component of these bonded sugar molecules can result in dire consequences and incompatibility with life itself. Changes in the pattern of glycans may lead to significant alterations in immune pathway signaling, as these glycans are important sugars that serve as the first contact point of cell to cell, but also host-pathogen interactions. Glycans are essential biomolecules for energy storage, system regulatory purposes, and play an essential role in various immune and inflammatory diseases. Their diversity exceeds that even of DNA, with up to 25,000 genes, RNA with approximately 360,000 microRNA, and every known cell is covered in glycans or sugars, which is as essential to life as the genome itself. Also, their structures exceed by orders of magnitude the number of proteins encoded by our genome, which can be up to 400,000 proteins. This makes glycoimmunology quintessentially supreme in all things health. Interactions include the discrimination of self and non-self, antibody recognition, pathogen binding, modulation of immunity, infectious disease, inflammation, the binding of hemagglutinin proteins, recognition of sialic acids, glycosaminoglycogen synthesis, metastasis, cellular adhesion, cell-to-cell signaling, intracellular transport, the elimination of xenobiotics, sphingolipid metabolism, and more. Glycan patterns initiate immune reactions, and virtually all cell surface receptors are glycoproteins. Glycan structures respond to environmental stimuli ranging from pH, ionic strength, hormonal stimulation, to inflammation, which aid in distinguishing health from disease, or self versus non-self. In a recent study titled Glycoimmunology, Ignore at Your Own Peril, they discussed the importance of this topic. In glycoimmunology, mannose, N-acetylglucosamine, and fructose, which is a seaweed polysaccharide, are at the core and the principal sugars found in human glycoproteins that impact our biology in a positive manner. Sialic acid is the Debbie Downer of the group. Sialic acid is the Debbie Downer of the group because it assists these stealth pathogens evade the innate immune response of the host, as sialic acid can hide mannose on the surface of the host cell and blocks mannose binding lectin, which we'll cover later. This prevents activation of the complement system, which is basically the adaptive immune response. Many viruses use sialic acid for binding to their target host cell, as sialic acid provides a good target for these viruses since they are abundant in large quantities on virtually all cells. Many pathogens also use glycan shields, or the glycocalyx as it's called, which is basically a protective sugar coat to evade host immune surveillance and pathogen recognition. If your immune system doesn't know a stealth pathogen's there to begin with, how is it going to mount a proper attack? By combining mannose, NAG, and bladderwrack together, it's like shining a black light in the darkness, revealing these hidden stealth pathogens, which allows our immune system to tag and bag them. Sialic acid compounds can interfere with the release of newly generated viruses from infected cells by inhibiting the viral enzyme known as neuraminidase. This is what skullcap and licorice do, among other compounds found in reverse effects. They block neuraminidase and subsequently impact sialic acid glycans, thus assisting in the blockage of the initial attachment and the release of viral particles from the infected cell as to stop viral replication. They also free up otherwise hidden mannose, so that mannose binding lectin may initiate the complement system. Reverse effects supports the body against viral transfection and interrupts this process. Hemagglutinin allows for viral attachment to the host cell, while neuraminidase cleaves or cuts the hemagglutinin off, which then allows for the virus to transfect or transmute to the next cell. Both skullcap and licorice have been shown to be viral neuraminidase inhibitors, thus shutting off the ability of the viral fusion or viral replication and eliminating the ability to travel to the next cell, which subsequently also assists in freeing up the hidden mannose.